Hi friends, today we are going to see the past year NEET questions for the chapter Cell, the Unit of Life, and today in this video we will cover part 8 and this is the last part for this chapter. So by now we've completed already seven parts for this chapter, so this part 8 is the last one for this chapter. So let's begin with the first question here. Read it here. This question was asked in the year 1991. Okay, fine, let's read it here. Addition of new cell wall particles among the existing ones. Okay, what do you call that as? Right? Fine. So you're given 4 options here. Option A, deposition, apposition, aggregation, intussusception. So, what do you mean by addition of new cell particles? It's the growth. Okay, they are talking in resemblance to the growth. Okay, so growth of cell wall occurs by 2 methods. Okay, addition is you're adding something, but at the same time, now for example, let's take an example here. Suppose if you say your kid is growing, she's adding on to her height, so it's growth. Okay, so likewise, addition of new cell wall particles amongst the existing ones is one way growth. And growth, if you see, of a cell wall is actually by 2 methods. Okay, is actually by 2 methods. Okay, so what are the 2 methods? One is intussusception, which is option C, and the other method is apposition. Okay, but the correct answer is intussusception because we are asking about the addition of new cell wall particles amongst the existing ones. Okay, therefore the option D is the right answer. Now what do you mean by intussusception? When the primary cell wall is stretched. Okay, understand here. When the primary cell wall is stretched. Okay. The materials of the secondary wall are secreted by the protoplasm deposited within it. Okay, so what happens here when the primary cell wall is stretched? You're adding on from the secondary part to the primary part. Now what is apposition then? Materials of the secondary wall secreted by the protoplasm are deposited in the form of thin layer, one above the layer. So apposition doesn't add on the existing ones, while intussusception adds on to the existing ones. Primary is actually starting from the secondary, it moves to the primary, so it is adding on to the primary part, secondary from the primary. Therefore the correct answer is option C, intussusception. So to your right side you can see whatever I explained is only on the right side. Okay, now next question. Now this question was asked in the year 1990. The latest model for plasma membrane is dash. Okay, I think all of you would have guessed by now the answer. Yes, it is fluid mosaic model. Option C is the answer. Fluid mosaic model, as you all know, is one of the most recent model of the biomembrane. Proposed by who? Proposed by Singer and Nicholson, which he proposed in the year 1972-74. So to your right side you can see, according to this model, the membrane does not have a uniform deposition. As we all know, they contain glycolipids, right? They contain glycolipids, then they will contain glycoproteins. So these are the things that are present. So you can see the picture here which shows the fluid as mosaic model. You have a extracellular fluid, you have glycolipid, glycoprotein. As I already said, the carbohydrate which is present in the minimal amount will get attached with the lipid and the protein, thereby forms a glycolipid and glycoprotein. So the recent model or the latest model of plasma membrane is a fluid mosaic model, which is option C. Okay, very easy question, right? Okay, now let's move to the next question here. This question again 1990, a very easy question again. We already discussed this in the earlier parts. If you remember, you can answer now. Hammerling's experiment of acetobutyria involved exchanging of dash. So this question, what is this acetobutyria? It's a green algae. Okay, it's a green algae. Okay, it's a green algae. Now, Hammerling he carried out experiments with this particular green algae. That is, he took 2 species from this Acetobularia. Acetobularia is a genus name, but under that he took 2 species and he carried out certain experiments. So here the question is, what exchange does it involve? Okay, here if you see, you're given 4 options here. You have nucleus, gametes, cytoplasm, rhizoid and stalk. It is obviously not cytoplasm and it is obviously not nucleus. It should be either C or D. Okay, see, as he took 2 species from the same genus, okay, of Acetobularia, he saw differences in shape of their caps, then he saw differences in the nucleus, okay, and many more other stalks. But what it involved, or what involved in exchanging, if you see, it is rhizoid and stalk. Okay, after removing the cap, if stalk of one species is grafted with the with the other one, okay, with the other species, shape of the cap will be determined by the nucleus and not by the stalk. So this experiment makes us clear that the characters of an individual are controlled by the nucleus. So what exchange does it happen? So what exchange did he do? He took 2 species, right, out of which one species, okay, one species here and one you have here. Okay, just remember, you take the root cap of here and you remember that you take the root cap. So when root cap is removed from one part, okay, he grafted from the other part and he found that the same kind of experiment, that is the same kind of root cap required for that particular algae, it actually growed. Okay, it actually started to grow. So what is understood here is, nucleus is what determines the character of the plant or direct character of this algae. So here, therefore, the correct answer is rhizoid and stalk. What did he exchange and carry out the experiment? It is rhizoid and stalk. The correct answer is option C. Next question. Now this question was asked in the year 1989. Okay, plasma membrane is made up of dash. Easy question again. I think by now you'd have guessed the answer. As we all know, plasma membrane is made up of proteins. What are they? Proteins, lipids and carbohydrates. Of course, they don't have nucleic acid. So therefore, the correct answer is option C, proteins, lipids and carbohydrates. Carbohydrates, if you see, they are covalently linked to both lipid and protein. That is why they form the constituent of what? Just now we saw in the earlier question when we saw fluid mosaic model. It forms glycoprotein and glycolipid. Right, very important actually. Though carbohydrate is present in minimal amount, the constituents are correctly matched and grown. Okay, now carbohydrate will be attached with protein and carbohydrate attached with lipid. Therefore, the correct answer is option C. Plasma membrane contains 3 constituents, proteins, lipids and carbohydrates will be in minimal amount. Next question. Now this question was asked in the year 1989. Polyribosomes are aggregates of dash. Again, I think it's easy question. Poly means many, polyribosomes. Okay, now as I already said, poly means many, right? Polyribosomes consist of about 4 to 8 ribosomes which are attached to a single strand of or a single string of mRNA. Okay, what do you mean by polyribosomes? What do you mean by polyribosomes? Poly means many. Okay, we already saw another example in the earlier parts where we talked about polytene chromosome. Several thousand strands of chromosome on a single DNA. Likewise, polyribosomes, if you see, many. So here if you see, what happens is 4 to 8 ribosomes will be present on the, or you can say several ribosomes held together by a single string of mRNA. Okay, this mechanism helps in the synthesis of several copies of the same protein. Why this synthesis is necessary? If you see, when we have to synthesize proteins of the same type, then we need this mechanism. Therefore, the correct answer is option D, several ribosomes held together by a string of mRNA. mRNA, what does M stand for here? M stands for messenger RNA. Okay, easy question in fact. Let's move to the next question. Okay. 1989 again. nucleoproteins are synthesized in. Now, what do you mean by nucleoprotein? You should know that, right? We already saw different types of proteins like what are the proteins? Tubulin, actin. We saw different proteins. Now, when we say nucleoproteins, they require another, they are proteins which are required for the nucleus. Okay, they are required for the nucleus. So, how can you describe nucleoprotein? They are a complex consisting of nucleic acid bounded to the protein. So, what is it? Nucleic acid bonded with a protein. Okay. Now, this nucleoprotein is synthesized. Is it nucleoplasm or nuclear envelope or nucleolus or cytoplasm? See, nucleoproteins, as I said, they are a complex protein which have nucleic acid attached with a protein. So, here they are conjugated proteins, or you can, the other name is also called as conjugated proteins. They are actually deoxyribonucleoproteins and ribonucleoproteins. So, where will they be synthesized? It is obviously not in the nuclear envelope, nucleolus or nucleoplasm. The correct answer goes to cytoplasm. Okay? The correct answer is cytoplasm of the cell. So, cytoplasm, I have given you a small diagram here which shows the cytoplasm here. What is cytoplasm? It is a fluid substance inside a cell. It includes everything inside the cell membrane except for the nucleus. Okay, the cytosol is the watery portion of the cytoplasm that where all organelles will float within the cytoplasm. Therefore, the correct answer to this question is option D, cytoplasm. Okay, easy question in fact. Next question. Again 1989, a bivalent consists of, right? A bivalent consists of. Now, when we say bivalent, bi means 2. Okay, we all agree with that. It's 2. So, either it should be one of the options here. Okay. Now, what is the common meaning of bivalent? Bi means, I said, it is 2. What is the common meaning of bivalent? See, whenever we talk about bivalent in biology, it always resembles homologous chromosomes. Okay, what does it relate to? Homologous chromosomes. Okay. We always refer to the homologous chromosomes associated in pairs, or you can say it's a pair of a homologous chromosome. Now, a bivalent consists of 4 chromatids, okay, and 2 centromeres. Okay, a pair of homologous chromosomes lying together in a zygotine stage of prophase 1 of the first meiotic division is called as bivalent. So, whenever we talk about bivalent, we always refer to the homologous pair of a chromosome. So, it will be formed during the prophase 1 of the meiotic division. Okay, so therefore, it will contain 4 chromatids and 2 centromeres. Understood? Therefore, the correct answer is option C, 4 chromatids and 2 centromeres. You always have to remember, whenever you have a question about bivalent, it always in talking with reference to the homologous chromosomes, and homologous chromosomes are during meiotic division. Only thing you need to know in extra here, in which phase will it occur, prophase 1 or what. So, here it is prophase 1. So, since it is prophase 1, it is 4 chromatids and 2 centromeres. Okay, next question. So, this question asked in the year 1988. Okay. According to the fluid mosaic model, plasma membrane is composed of, I think by now you have guessed the answer. Very easy question in fact. What is it? We all know it has phospholipids. Other than that, what else it has? See, fluid mosaic model was proposed by Singer and Nicholson in the year 1972. According to the fluid mosaic model, this plasma membrane, if you see, it is composed of phospholipids bilayer, we all know that, and a protein. Now, as we already saw, lipid is a main constituent. Okay, phospholipid is a main constituent of the fluid mosaic model. Other than that, you have proteins and carbohydrates are present in trace amount. Only it has to form a bonding between the lipid and the protein. Therefore, proteins which are present inside, which is present in the plasma membrane or the fluid mosaic model, they refer to the extrinsic and intrinsic part. Extrinsic means outside, and when I talk about intrinsic, it is inside. Okay, therefore the correct answer to this question is option D, phospholipids, extrinsic proteins and intrinsic proteins. This question is very easy. When you're learning, it is very important that you learn in depth. Always remember, whenever you talk about the fluid mosaic model, there are three constituents that you have to remember. What are the three constituents? Carbohydrates, proteins, lipids. You always have to remember this. The questions will be related to this. Okay, therefore the correct answer is option C, phospholipids, extrinsic proteins and intrinsic proteins. But carbohydrates, if you take, it will be only extrinsic. You don't have in the intrinsic part. Okay, proteins are only present in the extrinsic and intrinsic part, but carbohydrates are only present in the extrinsic part. Don't forget. Okay, next question. So this is the last question for this part 8 and the last question for this chapter also. Acetobularia used by Hammerling's nucleolacy. Okay, nucleolacy, cytoplasmic experiments is dash. So this question was asked in the year 1988. As we all know, acetobularia belongs to a green algae. So here, when you're carrying out a nucleocytoplasmic experiment, what happened? Acetobularia was used in that particular sequence. So here, acetobularia used in Hammerling nucleocytoplasmic experiment is a green algae, and this is actually a unicellular algae. Okay, so I've given you the answer partly. So it is unicellular, so it cannot be multinucleate. Very well, you have to understand, when we talk about unicellular organisms, the nucleus cannot be multinucleate. Therefore, the correct answer comes to option C. Unicellular, uninucleate green algae. Okay, the correct answer is option C, unicellular, uninucleate green algae. Fine. So to your right side, you can see a picture of how the acetobularia will be in overall appearance, physical appearance. Okay, this is how they will grow. Therefore, the correct answer is option C. Okay, so once again I'm telling you, acetobularia is a green algae which is unicellular and uninucleate. Okay, fine. With this we complete part 8, cell the unit of life. With this we complete all the parts for this chapter. We will meet in the next new chapter. Thank you.